Eukaryotic cells are far more complex than prokaryotic cells. One of their main characteristics is the compartmentalization. They possess a membrane-bound nucleus, as well as a number of membrane-bound organelles and an endomembrane system. And they are supported by an internal protein scaffold called the cytoskeleton. Let's begin by looking at the nucleus. The nucleus is the repository for all of the genetic information. Most eukaryotic cells possess a single nucleus. However, some possess more than one nucleus. This often results from fusion of multiple cells together, like we might see in muscle cell. The nucleolus is a region within the nucleus where we see a lot of ribosomal RNA synthesis taking place. In the figure to the right here, you can see the location of the nucleus and the fact that it's bound in a double phospholipid bilayer. So that is two bilayers of phospholipids. The cell itself is wrapped in just a single phospholipid bilayer. The nuclear envelope contains nuclear pores. These pores are small holes that control the passage of RNA and ribosomal components in and out of the nucleus. In eukaryotes, the DNA is divided into multiple linear chromosomes, as opposed to the circular chromosome that we see in prokaryotes. Chromatin is the normal form for DNA. This is when it's fairly unwound and is able to be used for protein synthesis. Chromatin is DNA wound around some protein cores, sort of like beads on a string, but it's easily accessible for transcription if the manufacture of certain proteins is necessary. Now, ribosomes are the cell's protein synthesis machinery. They are found in all cell types in all three domains, the eukarya, the archaea, and the bacteria. Ribosomal RNA is a protein complex, and it's synthesized in the nucleoid region of the nucleus. And because ribosomal RNA is indeed a protein complex, it requires messenger RNA as well as transfer RNA for its synthesis. Ribosomes can either be free-floating in the cytoplasm or associated with internal membranes. You'll see here a picture of a ribosome. The ribosome is composed of a large subunit and a small subunit. However, these two subunits don't come together until it's actually time to synthesize proteins. We'll investigate the synthesis of proteins in much more detail towards the end of this semester. Now, the endomembrane system is a series of membranes that's arranged throughout the cytoplasm, and it divides the cell into compartments where different cellular functions occur. This is one of the fundamental distinctions between eukaryotes and prokaryotes, as prokaryotes don't have any endomembrane-bound organelles. The components that we'll examine in this section involve the endoplasmic reticulum that's found continuous with the nucleus, and the Golgi apparatus, as well as the vesicles that transport materials between the two series of organelles. Let's look at endoplasmic reticulum. There are two types of endoplasmic reticulum. There's rough endoplasmic reticulum, shown here in blue, and smooth endoplasmic reticulum, shown in green. The rough endoplasmic reticulum, or RER, is rough because it has ribosomes attached to it. Remember, the ribosomes could either be bound or they could be free-floating. The free-floating ones are floating around in the cytosol, whereas ribosomes that are going to make proteins that are for export from the cell will bind to the rough endoplasmic reticulum. So these little dots are ribosomes attached all over the surface. So in short, the rough endoplasmic reticulum is also involved in synthesis of proteins and packaging of protein. The ribosomes will produce the protein and drop them off into the internal folds, the inner portion of this endoplasmic reticulum. And there, the proteins will be modified and packaged. The smooth endoplasmic reticulum has relatively few bound ribosomes and forms more of a tubular structure that you see here in the green. It has a variety of functions, like synthesis of phospholipids and storage of calcium, as well as detoxification. Now, where might you expect to see a lot of smooth ER? Remember, all of these different components in the cell vary depending on the cell's function. But what sort of cells might have a lot of smooth endoplasmic reticulum? Think of an organ you know of that's involved in detoxification. That's right, the liver will have tons of smooth endoplasmic reticulum. In fact, in individuals that take a lot of medications that need to be broken down in the liver, or after a night, say, of binge drinking, the amount of smooth endoplasmic reticulum increases markedly in order to assist us in the detoxification process. So again, the ratio of rough endoplasmic reticulum to smooth endoplasmic reticulum would depend on the cell's function. A cell that produces a lot of proteins for export, like let's say an insulin cell, would have a lot of rough endoplasmic reticulum, whereas cells that have a lot to do with detoxification or synthesis of phospholipids will have a lot of smooth endoplasmic reticulum. After a protein has been packaged in the endoplasmic reticulum for export, it's going to be shipped out to the Golgi apparatus. The Golgi apparatus consists of flattened stacks of interconnected membranes, or Golgi bodies. Because of this form, they appear a lot like endoplasmic reticulum. They function in packaging and distribution of molecules that are synthesized at one location and used at another, either within the cell or outside of the cell. For this whole system, I have an analogy. It's sort of like the endoplasmic reticulum is your office where you manufacture products with your ribosomes, and you prepare them for shipment. When you're ready for them to be shipped, you package them up in little vans and you drive them off to the post office or FedEx or UPS, whatever your choice of shipper is. FedEx, UPS, the post office, this is the Golgi apparatus. You drop off your packages on one side of the post office, and they will route them and label them in order to ship them. When they ship them out from the trans face or the transport face, the other side of the shipping center. So the Golgi apparatus has a cis face at which substances arrive, and then a trans face where substances are packaged off into their respective vans depending on where they're going to be transported to. Vesicles are these little membrane-bound sacs that the cell utilizes to transport molecules to a specific destination. So let's look at this whole system all as one. In the nucleus, we'll transcribe RNA from the DNA blueprints, and that RNA will go out and meet with a ribosome. Depending on whether the protein is to stay inside the cell locally or is for shipment outside of the cell, the ribosome will either make a choice to stay free and manufacture the protein in the cytosol, or bind to the endoplasmic reticulum in order to process the protein and put it into the endoplasmic reticulum. Once it's in the endoplasmic reticulum will modify the protein in whatever ways it needs. This is like your production center, packaging the materials up to be shipped out. The endoplasmic reticulum will bud off, because it is phospholipid bilayer, it will bud off small vesicles called transport vesicles. These are just little sacs of membrane-bound substance. So we have our protein of interest here that we're going to ship outside of the cell, and this protein will be transported through the cytoplasm and dropped off at the Golgi apparatus, or you dropping your materials off at the packaging and shipping center. Inside the Golgi apparatus, much like inside the UPS plant, your packages will be labeled and sorted and modified for shipment to their destination. Once all of these processes have occurred, another vesicle will bud off that contains your packages, and it will be shipped out and bind with the cell membrane and drop the contents out into the extracellular fluid, where they could be picked up by the blood and transported to other regions of the body. This is a great figure for you to summarize the endomembrane system. Take a moment here to stop and diagram the endoplasmic reticulum, transport vesicles that move materials from the endoplasmic reticulum to the Golgi apparatus, Diagram the Golgi apparatus, and then the transport vesicles that transport things away. Be sure to include the cis phase and the trans phase, as well as a notation of ribosomes that are bound or free. Be sure to explain each of their different functions. One final component of the endomembrane system are lysosomes. These are membrane-bound digestive vesicles. They're basically specialized vesicles that are filled up with enzymes that catalyze the breakdown of macromolecules and other substances. They arise from the trans phase of the Golgi apparatus, and they'll fuse with either old cell organelles and digest the old cell organelles, or they'll fuse with other vesicles that have been engulfed by the process of phagocytosis. Once they fuse with the food vesicle, they're able to digest the components within it. The digestive enzymes in the lysosomes are optimally active at acidic pH. Lysosomes are activated when they fuse with the food vesicle or with the worn-out organelle. The fusion event activates proton pumps in the lysosomal membrane, which lowers the pH inside the lysosome. And then the digestive enzymes are turned on and they can degrade macromolecules of the food that's in the vesicle or utilize them for destruction of the old organelle. There's a number of human genetic disorders collectively called lysosomal storage disorders. Tay-Sachs is an example of one of these. It's caused by the loss of one lysosomal enzyme. And this is the enzyme that's necessary to break down membrane glycolipids that are found in nerve cells. Accumulation of this glycolipid in lysosomes affects nerve cell function. This leads to a variety of clinical symptoms, such as the seizures and muscle rigidity that we see in Tay-Sachs. So basically, the lysosomes are broken, and because the lysosomes are broken and unable to degrade the contents fully, they just become packed with trash. And the nerve cells fill up with lysosomes full of products that they're unable to break down. Tay-Sachs is a horrible genetic disorder. Another variety of enzyme-bearing membrane-enclosed vesicles are the microbodies. There are many different types of microbodies, but the peroxisomes are of particular interest. They contain enzymes involved in the oxidation of fatty acids. If these oxidative enzymes weren't isolated within the microbodies, they would tend to short-circuit the metabolism of the entire cell because of the peroxide that's produced as a byproduct. Of course, inside the microbody, this is rendered harmless by catalase. So microbodies work in a very similar way that lysosomes work. They fuse with anything that has a lot of fatty acids that need to be oxidized or broken down. Vacuoles are final membrane-bound structures, and they're only found in plants. There are a number of different vacuoles, the most prevalent of which is the central vacuole that fills the center of the cell. The central vacuole in a plant cell functions in its tonicity, its rigidity. When this central vacuole is full, it's filled with fluid and it keeps the cell turgid or solid so that the plant can stay upright. There are two other types of vacuoles. The contractile vacuole that we see in some protists that allow it to move around, and then storage vacuoles that we might see in like a potato where plants store lots of starch. Let's take a quick card quiz before we finish this section. Which of the following organelles is not part of the endomembrane system? Which organelle contains digestive enzymes that are used to recycle unwanted material in the cell? The synthesis of proteins takes place and blocking ribosomes would prevent. In Tay-Sachs disease, lysosomes accumulate lipids and burst. How will this affect the cell? Now let's take a look at the card quiz answers. Yellow, green, red, blue. If you have something different than this, then go back and investigate. Or maybe I got one of them wrong. In which case, get on the discussion board and question it.